Chapter 12, Neural Tissue. The nervous system is made of neural tissue, and the neural tissue is composed of neurons, which are the actual nerve cells that conduct the nerve impulse, neuroglial cells, which are supporting cells, and these are going to help protect and nourish the neurons, insulate them, and keep them healthy. They outnumber the neurons by quite a bit. Also in the nervous system are the organs, the brain, the spinal cords, the nerves, and the receptors in your sense organs. And the nervous system function is to provide a swift, brief response to stimuli through a temporary modification in the activities of the organ systems. It is not supposed to be for a long-term response. The long-term response will be picked up by the endocrine system. The nervous system has two parts, the central nervous system and the peripheral nervous system. The central nervous system, also known as the CNS, is composed of the brain and the spinal cord, and its function is to process any incoming sensory information and send out motor commands to the organs. The brain is the site of memory, intelligence, learning, and emotions. The peripheral nervous system, the PNS, is all the neural tissue that's outside the CNS. This is going to deliver sensory information to the CNS and carry motor commands to the peripheral tissues and systems. Your nerves are made up of bundles of neuron axons. These are the nerve fibers. These are going to carry the sensory information and motor commands in the peripheral nervous system. Peripheral nerves are composed of nerve fibers plus the blood vessels and connective tissue that are associated with it. Okay. And under peripheral nerves we have two types. The cranial nerves are the nerves that are connected to the brain running out to the body, and the spinal nerves are attached to the spinal cord and run out to the body. Peripheral nervous system has two subdivisions. The afferent division, with an A, brings sensory information to the central nervous system from receptors that are in the peripheral tissue and the organs. The receptors are special structures that detect changes in the internal or external environment or respond to some kind of specific stimuli. Three types of receptors are somatic sensory receptors. They monitor skeletal muscles, joints, and skin. The visceral sensory receptors, which monitor your internal organs, and your special sensory receptors that are monitoring your special senses of sight, taste, smell, and hearing. The second division of the peripheral nervous system is the efferent division, with the letter E. This will carry motor commands from the CNS to the effectors. Effectors will respond by doing something. So we'll carry motor commands away from the central nervous system. Your effectors are going to include skeletal muscle, smooth muscle, cardiac muscle, and your glands. We have two subdivisions under the efferent division, the somatic nervous system. This is going to control your skeletal muscle contractions, and the autonomic nervous system, which is going to control your smooth muscle, cardiac muscle, and glandular secretions. Okay. The autonomic nervous system, which is an automatic regulation, can be further subdivided. It will control the same organs, but it will control them under different circumstances. The sympathetic division will control them under stressful conditions, and it's referred to as the fight or flight division. And the parasympathetic division will control those organs under relaxed conditions, and this is called the rest and digest division. Both are controlling the same organs. They're antagonistic to each other. The neuron is the cell of the nervous system that conducts the electrical impulse. Okay, there are certain characteristics that all neurons have in common. They all have a high metabolic rate, which means they need a lot of oxygen and glucose to keep working. They have longevity. They will last for your lifetime, and they are amitotic. No cell division occurs, so if you damage a neuron and it dies, you do not replace it. The general structure of the neurons is going to be broken into four general characteristics, and we're going to be looking at a multipolar neuron, which is the most common type of neuron for those characteristics. First, we name the cell body the soma. This is where a typical cell's organelles are going to be found. A typical cell will, like we learned when we did cell structure, you would think of as the cell body, the soma. Off that soma there are going to be extensions. One set of extensions are called the dendrites, and these are slender processes, and they're going to receive the incoming information. The dendrites will break down into even smaller processes called dendritic spines. On the other side of the soma, there's going to be one single extension that is a long cytoplasmic extension called the axon. And the electrical impulse, once it's generated, will travel down the axon. The end of the axon is branched into telodendria, and the telodendria will then touch other neurons or other cells and pass the electrical impulse to the next cell in line. Now the neuron structure in more detail. The soma, the cell body, has a large round nucleus which has a nice large round prominent nucleolus inside it. Remember, your nucleolus's job is to make ribosomes. So we have a large amount of ribosomes within the soma because we have a large amount of protein needs, which is the job of the ribosomes. Okay. The cytoplasm that surrounds the nucleus in the soma is called the perikaryon, which means a round nucleus. And the cytoskeleton is made up of neurofilaments and neurotubules. And bundles of neurofilaments are put together and called neurofibrils. And these bundles of neurofibrils will extend to the dendrites and the axon to give it some internal physical support. There's a lot of mitochondria because we have a huge amount of ATP needs in our neurons. Right, back to your ribosomes. We have a lot of them. They're both free and attached to the rough endoplasmic reticulum, and there's so many that they actually are grouped together in one name under Nissl bodies. And the Nissl bodies give the soma a dark coloration because it's so dense with the ribosomes. When you're looking at the central nervous system, we have both white matter and gray matter. The gray matter of the brain and the spinal cord is where the somas are that are filled with those Nissl bodies. Neurons lack centrioles, because they can't divide. They don't do cell reproduction. There are few neural stem cells in the adult that are active, and those are going to be found in your nose to regenerate the receptors for smell, and in the hippocampus, the part of the brain involved with memory. The dendrites are the highly branched extensions that receive incoming information that end in the dendritic spines as smaller branches. So you want to think of this sort of like a tree with the branches sticking up into the sky, and if you get struck by lightning, that lightning would hit the branches first and then go down to the cell body. Okay. The axon is on the opposite side of the dendrites, and this is a long cytoplasmic extension. It goes from the soma down to end in small branches called telodendria. Now, this is where a lot of the action is going to happen that we're talking about in the axon, so we give special names to the parts of the axon. The membrane of the axon is called the axolema. Okay. The axon itself has cytoplasm within it, and the cytoplasm is called the axoplasm. Now, when you're looking at the axolema, where the cell body and the axon meet, there's a little widened area that's called the axon hillock, and then right below that is the initial segment of the axon, and then the information coming in, the electrical information will come in, add up at the axon hillock, and then start traveling down the axon to the telodendria, where it will be passed to the next cell in line. Now, your axoplasm, it's the cytoplasm. It's going to have lysosomes, mitochondria, some neurofibrils, neurotubules, but no ribosomes, and most of the other organelles are not found in the axoplasm. The telodendria are the thin branches of the axon, and they're going to end in a nice little bulbous rounded end called synaptic terminals, often referred to also as axon terminals. Okay. Your axons may be wrapped in an insulating material will make up the myelin sheath, and the function is to protect the axon and speed up the rate at which that action potential, the electrical impulse, will travel down the axon. Once that electrical impulse makes its way down the axon, it has to be passed to the next cell in line. And this process is called a synapse. This is when the neuron communicates with another cell. There's going to be two cells involved in this process. The presynaptic cell, the one before the synapse, which is sending the message, and that's usually a neuron, and the postsynaptic cell, which is the one after the message is sent, and it receives the message. This can be either a neuron, a muscle, or a gland. Okay, now often we are going to communicate with neurotransmitters, where the presynaptic neuron releases these chemicals, neurotransmitters, and they make their way across to the postsynaptic cell, and they give them information. These are going to be, your neurotransmitters are going to be found packaged in synaptic vesicles at the end of those, inside those synaptic terminals. Now there are different types of synaptic terminals where the synapses occur, okay, depending on what the cells are that are involved and how complex the cells are. A synaptic knob is going to be found at the end of the teledentary. It's just a rounded packet, and this is going to be found when the synapse is between two neurons. The neuromuscular junction is a synapse that occurs between a neuron and a muscle cell, and a neuroglandular junction is a synapse between a neuron and a gland cell. We're going to have to move some materials down from the soma to the synaptic terminals, and they're going to have to move along the axon through the axoplasm, the cytoplasm. Okay. This movement can occur in both directions. Antegrade flow is when materials made in the soma are going to be moved down to the synaptic terminal. The materials are going to be things like enzymes and neurotransmitters that were produced up in the soma. We also have retrograde flow, where material will move from the synaptic terminal back up to the soma. Things like debris and waste products will have to be moved up to the soma so they can deal with it and get rid of the materials. Now there are some viruses that have learned to use this retrograde flow as a sneaky way to enter the body. Rabies, herpes, polio, they will all sneak in the unguarded back door into the synaptic terminal, make their way up the axon through retrograde flow, and then enter into the central nervous system to then start attacking the cells. There are different types of neurons, and they're going to be classified by structure, which is anatomy, and by function, physiology. Structural classification is based on the relationship of the dendrites to the cell body and the axon. The first type is called an anaxonic neuron. Now, if you put the word "an" in front of any word, it means without. So without axons. This is a small neuron, and it has extensions off it that all look like dendrites, but one of them is the axon, so we can't tell which one is the axon. These are relatively rare, occurring in the brain and some special sense organs, and their function is poorly understood. The second type of neuron is a bipolar neuron, meaning two extensions off the cell body. One will go off one side and give rise to the dendrites, the other will be the axon and give rise to the synaptic terminals. Okay. These are also relatively rare, and they're found in our special sense organs. Okay. They're smaller than the unipolar or multipolar neurons, the next two to talk about. Unipolar neurons have only one extension off the cell body. So when you look at this, you want to look at the cell and look at it looks like a T formation. One extension that then branches to two sides, and one side of that branch will give rise to the dendrites, and the other will give rise to the axon. Okay. Your unipolar neurons are most of your sensory neurons of the peripheral nervous system. Okay. Multipolar neurons, the most common neurons, and the one we use for the general neuron structure, has two or more dendrites off the cell body and one axon. It's the most common type in the peripheral nervous system. All motor neurons that control skeletal muscles are multipolar, and most of the CNS neurons are multipolar. About 99% of all the neurons in the body are multipolar. Functional classification of the neurons allows us to divide the neurons by what their job is. We have sensory neurons, These are going to make up the afferent division, that starts with an A, of the peripheral nervous system, and they're going to deliver information from your sensory receptors to the central nervous system. Most of these are unipolar neurons. The axons make up the afferent fibers of the nervous system, and there are about 10 million different sensory neurons. They will be divided into two subgroups: somatic sensory neurons, which are monitoring your external condition of what's going on outside the body and in the skin and the joints, as well as visceral motor neurons, which are monitoring your internal conditions in the body. Your sensory neurons get their incoming information from sensory receptors. These are little either specialized sensory neurons or cells that are going to be monitored by the sensory neurons. And there are three types. Exteroceptors are getting information about the external environment, such as touch, temperature, pressure, sight, smell, hearing. We have proprioceptors, which are specialized stretch receptors that monitor your skeletal muscles and your joints. And then we have interoceptors, which are monitoring the viscera, the guts, the digestive, urinary, cardiovascular systems. They also are going to be involved with taste, deep pressure, and pain. Under functional classification, the second type of neuron is the motor neuron. These make up the efferent division of the peripheral nervous system. They're going to carry instructions from the central nervous system to your peripheral effectors that are in your peripheral tissues, your organs, okay, and the organ systems. These are mostly multipolar neurons. Okay. Their axons make up the efferent fibers, and there's about 500,000 motor neurons. Again, we're going to subdivide this. Somatic motor neurons are found in the somatic nervous system. They control your skeletal muscles. Your visceral motor neurons are found in the autonomic nervous system, and they control smooth muscle, cardiac muscle, your glands, and your adipose tissue. The third type of neurons are called interneurons, and these are going to be found in the central nervous system, in the brain and the spinal cord. And they're going to be responsible for distributing the sensory information to the central nervous system that's coming in on the sensory neurons, and then they're going to turn around and coordinate the motor activity and send that information, once they've decided what they're going to do, out on the motor neurons. There's going to be one or more interneurons between a sensory and a motor neuron, and the more complex the response is to a certain stimuli, the more interneurons there will be. Okay. They're involved in all your higher functions, such as memory, learning, and most of these are also multipolar neurons, and there's about 20 billion interneurons making up the central nervous system. The neuroglial cells are going to be the cells that support and protect and insulate the neurons. They're found in the central nervous system and the peripheral nervous system. In the central nervous system, we have 4 different types of neuroglial cells, and they outnumber the neurons 10 to 1 and about half the total mass of the brain. The central nervous system neuroglial cells start with the ependymal cell. Now, inside your brain you have ventricles. Ventricles are fluid-filled sacs. You also have in the spinal cord a central canal, which will also have fluid. Okay. Lining the ventricles and the central canal are the ependymal cells, and they're going to assist in producing, circulating, and monitoring the composition of your cerebrospinal fluid. Your cerebrospinal fluid is one of the main fluids of the body that we use for analysis. It's right up there with blood and urine for looking for any kind of problems, infections, any kind of inconsistencies. Now, the cerebrospinal fluid is in the ventricles, the central canal of the spinal cord. It also surrounds the brain and the spinal cord, and its function is to provide a protective cushioning around the brain and spinal cord. It will transport dissolved gases, nutrients, and waste products. The ependymal cells have some cilia associated with them, and they use this to circulate the CSF, cerebrospinal fluid, which we are constantly moving and remaking and absorbing. The second type of neuroglial cells in the central nervous system are called astrocytes. They look like star cells, so that's where astro comes from. These are the largest and most numerous of the neuroglial in the central nervous system, and they have a number of different functions. One of the most important is that they maintain the blood-brain barrier. Now, the blood-brain barrier, in your brain, you don't want everything getting out of the blood like it can in other places of your body. If you have drink a cup of coffee and the pH is off, you don't want the pH to get out and damage the neurons. They're highly sensitive. So we coat all the blood vessels and the capillaries with extensions that come off the astrocytes. The astrocyte looks like it's got little extensions like feet coming off it, and they got flat little pancake extensions, and they wrap those pancake-like tissue around the blood vessel. And one blood vessel will have numerous little wrappings on it all the way down, so it's completely covered with this blanket of material. An astrocyte will reach out and touch a number of different blood vessels, and one blood vessel will be wrapped by a number of different astrocytes. This is also going to provide a little bit of structural support, and stabilize the blood vessels, which is the second function. Structural support, these extensions are filled with microfilaments, they produce a cytoskeleton, and they extend throughout the whole brain, holding on to the blood vessels, and they fill up the brain with a large amount of material, and that provides a huge amount of structure. They're involved with damage repair. If your neural tissue gets damaged, you get a hole in your brain. You can't make new neurons, but you don't want the hole to stay there. So these just kind of fill it up and put like a filler in there and form scar tissue so that there isn't an existing hole anymore within the brain. The fourth function is that they glide the neurons during development. In the embryonic brain, these are going to be involved in interconnecting the neurons, making sure one neuron grows in the right direction so it will ultimately hit the next neuron. Otherwise, they will have trouble talking to each other. And the last thing they do is they control the interstitial environment around the neurons. That's the fluid that is around the neurons. They're going to be involved with regulating how much sodium, potassium, carbon dioxide is out there. They're going to reabsorb anything that's excess. They're going to move nutrients and ions and dissolved gases between the capillaries and the neurons. Control blood flow through the capillaries, so if you need more oxygen, you'll increase the blood flow and pick up more oxygen. They're going to be involved in absorbing and recycling neurotransmitters, and they will release chemicals that will either enhance or suppress communication across the synaptic terminal. The third type of neuroglial cell in the central nervous system is called the oligodendrocytes. These are going to produce a material that will wrap around the axons of the neurons and will cause them to be insulated. The material is called myelin, the myelin sheath, so they myelinate the axons. Now, the oligodendrocytes have a number of little hands that reach out, again, extensions, and they will touch a number of different neurons, and each neuron will be wrapped by many oligodendrocytes. They will provide this wrapping that then leaves a little space between the layers of insulation, and these spaces are going to allow the action potential to jump from one space to the next space and move down the axon. The spaces are called nodes of Ranvier. Okay? And in the white matter, the central nervous system is dominated by these myelinated axons. They also are going to help to provide some structural framework for the central nervous system. Your microglial cells are the fourth and final of the central nervous system glial cells. These play a role in defense. They're removing waste and debris and pathogens. They are related to the stem cells that gave rise to the macrophages, which were the large phagocytic cells, white blood cells. Okay. And they're busy working all the time because the cells of the immune system can't enter the central nervous system because of that blood-brain barrier. So there's nobody in there but the microglial to do defense. There are two types of neuroglial cells in the peripheral nervous system. The first type is called satellite cells, and these are going to surround the neuron cell bodies and isolate them from the surrounding tissues. They have a similar function to the astrocytes. They regulate the environment around the neurons by regulating nutrient and oxygen and carbon dioxide content, as well as neurotransmitter levels. If you think about satellite cells are up in the sky and astrocytes are star cells, so try to remember the two of them together. Satellite from the peripheral nervous system, astrocytes from the central nervous system. The second type of glial cell is the Schwann cells. These are going to myelinate the axons in the peripheral nervous system. Therefore, they have a similar function to the oligodendrocytes in the central nervous system. Okay. The difference here is that one Schwann cell will flatten out like a pancake and wrap around one part of a single axon. Then there will be a space, and then there will be another Schwann cell. Then there will be a space and another one. So they don't touch other axons, which was the case in the central nervous system. Again, the spaces where there is bare axon are called the nodes of Ranvier. Okay. The functions of the Schwann cells are going to be to insulate the neurons, and this insulation is going to allow the action potential to travel quicker down the axon. They're also going to be involved in repair process after injury to a neuron, and they are involved with holding bundles of unmyelinated axons together. This diagram is showing a neuron in the peripheral nervous system with the axon myelinated by the Schwann cells and the little spaces between the Schwann cells, the nodes of Ranvier, where the action potential will jump as it runs down the myelinated axon. This figure shows the group of unmyelinated axons bundled together and surrounded by the Schwann cells, insulating the axons. This is how a neuron will respond to injury. In general, the response to any kind of damage is limited, and it's a very stereotyped response. In the cell body, the Nissl bodies will disperse. Those are all the ribosomes. The nucleus moves towards the side of the cell, and protein synthesis increases rapidly. Now, the key to recovery is the extent that the axon was damaged. If it was only pinched for a little short time, it should be able to recover within a few weeks. If it's pinched for a really long time, it actually may damage it completely, and the distal end will be severed. Okay? And therefore you will not recover. In the peripheral nervous system, what is going to happen when you have damage is that the axon distal to the injury, wherever the injury occurred, starts to degenerate and break down. The macrophages are going to arrive to start cleaning up the debris, because they can get in here now because it's been exposed to the environment. Okay, and the Schwann cells that are wrapping and making the myelin sheath don't disintegrate or degenerate. What they do is they proliferate. They make a lot more, and they go right down that path where the neuron had gone and form a little core. This is so it can follow the path when the neuron does start to recover, so one neuron will still be able to come in contact with the second neuron in the path. Okay. The axon will grow along the path, and then as it grows, the Schwann cells will wrap it again with the myelin sheath, and eventually it may reestablish normal function. Now, if the damage is in the central nervous system, it's much more limited, mainly because there are more axons involved. So the damage involves a huge amount of axons. And the astrocytes immediately start producing scar tissue, so the axon can't grow in the damaged area, and they release chemicals that block the regrowth of the axons. Okay. A neurophysiology overview. We're going to look at how the neuron functions. First definition we need to listen to is the transmembrane potential. This is an electrical difference across the cell membrane. So it'll have an electrical charge inside the cell and a different electrical charge outside the cell. This transmembrane potential is going to change as the cell moves from rest to an actual electrical impulse. So we're going to start with our resting potential. This is the transmembrane potential of a neuron at rest. This potential has got to change so that neural activity can occur. We have graded potential. This is a temporary localized change in the resting potential. And this potential will decrease as it moves farther from the stimulus. Now, this is to demonstrate this, it would be like if I had said something quietly to the first row of students who then didn't tell anybody else and nobody else in the room heard it. So I said it and then it decreased slow and they heard it, but then it was so low that nobody else could hear it. The next potential is called the action potential, and this is the actual electrical impulse. And this is going to be propagated across the membrane and down the axon, the whole axon, to the synaptic terminal. Your action potential does not weaken as it moves away from the source. It travels along to the synapse, and the AP is produced by many graded potentials if they are strong enough and they add up so that you can reach the minimum potential for an action potential to begin. Now, whereas the graded potential, I said, think of it as me saying something quietly and only the first row of students heard it. An action potential is I would say something loud enough the first row heard it, then the first row repeated it the same strength so the second row heard it. Then the second row of students repeated it again. So it keeps traveling through the whole class. It doesn't get any lower in volume and moves all the way from the front to the back of the room. That's the difference between the graded potential and the action potential. Now the action potential is started, it runs down the whole axon and gets to the end where the synaptic terminals are, and we have synaptic activity. This is where the potentials from the presynaptic cell are going to strike the postsynaptic cell and stimulate graded potentials on the postsynaptic cell. Often this involves the release of neurotransmitters, and they move across the synaptic cleft, bind with the receptors on the postsynaptic membrane, and start the graded potentials. And the last thing is informational processing. The postsynaptic cell is getting hit by numerous, up to thousands of different synapses at the same time. They may each give a little different graded potential, so the postsynaptic cell has to add up all those graded potentials that are coming in and then determine how it's going to respond to that information. Is it going to be enough to generate an action potential, or does it fall short and no action potential starts? At rest, the neuron and axon have a certain characteristics of what's inside the cell and what's outside the cell, and we're going to look at ion concentrations. Inside the cell, in the soma and the axon, you are going to have a large amount of potassium ions, which are positively charged, and some large negative proteins. Outside the cell, you are going to have positively charged sodium ions. Now, the potassium and the sodium are both small enough ions that they can sneak through small little leak channels. The leak channels are specific to the ion, so there's a potassium leak channel that will allow the potassium to diffuse from inside the cell to outside the cell. This is a passive process, going with the concentration gradient using no energy. And there will be sodium leak channels that will allow the sodium to diffuse into the cell. Again, passive, no ATP being used. Okay. Also outside with the sodium is the chlorine ions, but they aren't really involved in the process. Okay. Now this transmembrane potential is going to be the electrical difference across the cell membrane. Inside we have positives plus these big negative proteins that can't leave the cell. Outside we have mostly positives with some smaller negative chlorine items that don't add up to much. So when you look at the electrical charge, the inside of the cell has a negative 70 charge. And the unit we use for this is millivolts, MV. So inside the cell there is a negative 70 millivolt charge. This is the resting potential, the transmembrane potential of the resting cell. This must change for neural activity to occur, but we always have to get the neuron back to the resting potential to start a new neural activity process. Now we have to maintain this resting potential. If we lose the resting potential, we're going to have a problem and the neuron won't be able to work. So even though the potassium is sneaking out constantly through the leak channels and the sodium is sneaking in constantly, we need to pump the potassium back in and the sodium back out so we keep the concentration gradient correct with sodium outside and potassium inside. We are going to use the sodium-potassium exchange pump, or ATPase pump. This is an active process. This is going to go against the concentration gradient, and it's going to use ATP. So this is a constant battle. Sodium is sneaking in, and then it's being pumped out. Potassium is sneaking out, and it's being pumped back in. And the exchange pump brings in 3 sodium ions for every 2 potassium ions it pumps back out of the cell. Recap of resting potential. Inside the neuron, high potassium and negative proteins, resulting in a negative 70 millivolt charge, which is the transmembrane potential. Outside, you have high sodium concentration and some chlorine ions in the interstitial fluid. We have to be at resting potential for the neuron to get excited and start an action potential. In addition to the passive leak channels we have on the axolemma, the membrane of the axon, there are also some active gated ion channels. Now these active gated ion channels are going to open and close in response to a specific stimuli. They have 3 different states that they can exist in. First, they will be closed but capable of opening. Then once they're open, they are considered activated. And as soon as they close, they are incapable of opening and are considered inactivated. This state is going to occur immediately after closing the activated gate, and after a bit of time and the cell returns to its resting potential, it will be capable of being opened again. Now, there are 3 different classes of gated channels which are separated by the stimulus that triggers them to open. Chemically regulated gated channels will open when a specific chemical binds on the receptor of the gate, and that will cause it to open. An example of this was acetylcholine on the gates of the skeletal muscle. Okay? And these are going to be involved in synaptic communication, the chemically regulated. Voltage regulated gated channels are going to be found in areas of an excitable membrane, and they are going to respond when there is a change in the transmembrane potential. Okay. Examples of this are going to be your sodium, potassium, and calcium channels. Each channel is for a specific ion and works when due to the diffusion of the ions. The third is the mechanically regulated gated channels. These are going to respond to physical distortion of the membrane surface. They're going to be important in sensory receptors such as touch and pressure. Okay. Now, at rest, when you're at the resting potential, the gated channels are closed. They will open, and then once they do open, the ions that they're specific for will run through the channel rapidly and change that transmembrane potential. So moving it from a resting potential to another. The potential that they will stimulate once they open it is called a graded potential. That is the change in the transmembrane potential that cannot spread far from the stimulus, so it doesn't move too far from that gate. Okay. When the sodium gates open, they'll allow sodium to enter into the axon, changing the transmembrane potential and causing it to become less negative, because we're moving positive sodiums into that negative 70 millivolt environment, so it's going to get less negative. Okay. That will possibly, move the potential now from negative 70 maybe to negative 68 or negative 66. Any single graded potential, one graded potential, is usually not strong enough to start an action potential. But if you get a number of them in there, they can add up and start an action potential. A few more definitions before we can start with the action potential. First, when we talk about polar, we're talking about the poles, like a positive and negative pole on a battery. Okay? We do depolarization. That is when there's a change in the resting potential and it becomes less negative to even becoming positive. Okay? So if we started at negative 70 millivolts, and these numbers are important so you can see how they change, negative 70 millivolts is the resting potential. If the resting potential moves to negative 69, it has undergone depolarization. If it moves all the way up to positive 30 millivolts, it has undergone depolarization. And this is due to the fact that those positive sodium ions will move inside the axon And then they will also be carried down by the local current, which is just a movement of positive charges parallel to the inner and outer surface of the membrane. So it's like think of a current in a little river. It's being dragged downstream and making the next area a little less negative, and that will depolarize the next portion. The degree of polarization will decrease as you move farther from the source. Repolarization is when we're going to go back, if we've made it less negative or positive, and we now want to bring it back to the resting potential. Okay, repolarization occurs after depolarization has happened. So now we're going from our positive 30 millivolts back to positive 29 millivolts, all the way up to negative 70 millivolts, the resting potential. Okay. Hyperpolarization is when the cell becomes even more negative than the resting potential. So it goes from negative 70 millivolts to negative 80 millivolts. Okay, this will actually happen once we depolarize and we're repolarizing. We don't close all the gates quick enough, so we let in too many of the positive potassiums, so it overshoots the negative 70 and hits negative 80. Then we gotta get rid of some of those to bring it back down to the negative 70, to the resting potential. Now we're going to look at starting at resting potential and then moving through the action potential. Now, at resting potential, sodium is outside. The potassium is inside with the negative proteins. This gives a negative 70 millivolt charge inside the axon. The voltage-gated sodium channels and potassium channels are both there, but they are closed. So in this figure, the purple is for the is for the sodium, and the orange colored is for the potassium. The first thing we have to do to generate an action potential, which is the electrical impulse that we want to have traveling down the axon, is we have to depolarize the axon to what's called the threshold potential. The threshold potential is going to be the minimum amount of depolarization needed to open those voltage-gated sodium channels. And that will, when they open, that will start the action potential. So we have our leak gates for sodium, and they're leaking the sodium into the axon, and this will trigger graded potentials. And if enough graded potentials happen and enough sodium is leaking in, that those graded potentials add up to negative 60 millivolts, then the cell has hit the threshold potential. And when the threshold potential is hit, that will trigger the opening of the voltage-gated sodium channels, and then the sodium will rush into the axon. So we go from negative 70 millivolts to negative 60 millivolts, hitting threshold. That opens up the sodium gated channels, and then the sodium charges in, and that will then take the electrical impulse into the positive range. Now your nerves are going to respond with what's called the all or none principle. And this is that if a given stimulus will either trigger an action potential or nothing at all, then all action potentials are of the same strength. So if the stimulus will bring it to the axon to threshold, then that will start an action potential. And any stimulus that brings it to threshold will start an identical action potential. If the depolarization only takes it up to negative 61 millivolts, not negative 60, which is threshold, then no action potential will be triggered. If it took it from negative 70 to negative 69 or negative 70 to negative 61, it's the same response, nothing happened. Once you hit negative 60, boom, the action potential is off and running. And even if you go much higher than that, it doesn't change the action potential. Once we open those voltage-gated sodium channels, the result is rapid depolarization, because all these positive sodiums are charging into the cell. And it's going to go from negative 60 millivolts all the way up to positive 30 millivolts in less than 1 millisecond. Okay? Remember, your action potential started at plus 60 millivolts, the threshold, so it's already going and running down the axon. And depolarizing the membrane past the negative 60 millivolts has no effect whatsoever on the action potential. It's gone, and negative 60 is the magic number when it gets started. When we get to that positive 30 millivolts, we're going to inactivate those voltage-gated sodium channels. We're going to close them. Okay? And at the same time, we are going to open or activate the voltage-gated potassium channels. Okay? When it hits positive 30, the sodium channels close, the potassium channels open. Because as soon as we have hit that negative 60 millivolt threshold and we've started the action potential, the rest of the time we want to start to clean it up. We can't immediately close the gates, so the sodium runs out real quick, it gets to plus 30. Now we slam those gates closed so no more sodium can get into the cell. Okay? And now we have a little bit of a mess. We look at our cell now, and now we have sodium and potassium inside, and we have a positive 30 charge. We wanted to have sodium outside, potassium inside, and a negative 70 millivolts at resting potential, and we have to get back to that at some point. Okay? So we're going to first try to fix the imbalance in the electrical charge. The only way we can do that, to go back to a negative, is to get rid of some kind of positive charged molecules or ions. Now the only way sodium gets out is it's going to be getting out through that sodium-potassium exchange pump, and that's a slow process. So we can open the gates for the potassium, which at this point all we care about is that they're positively charged ions, and we can let those potassiums run out of the cell. So we're throwing out a bunch of positives, so we can actually get back to the negative 70 millivolt charge. Once we hit negative 70, we want to close all those potassium gates so that we don't affect the charge anymore. But we're a little bit slow on that, so there's a little bit of a time lag, so a little bit more potassium gets out than we want, and it actually will take the charge, the potential inside the cell, from negative 70 all the way up to negative 80 or even negative 90 millivolts. This is when we have hyperpolarized the cell. So we're going to, at negative 90, because it took us a little too long to close those potassium gates, we close those gates. At this point, both the sodium and potassium gates are closed. The sodium is inside the cell, the potassium is outside, and we're at negative 90 millivolts. Well, we're going to slowly get back down to negative 70 millivolts. But we have a big problem now, is that the sodium and potassium are on the wrong side of the membrane. So we're going to have to work on that. And for that, we're going to use that sodium-potassium exchange pump, which will take a while to slowly pump out the sodium and pump the potassium back in. But there's enough sodium and potassium in other places that it doesn't slow down the action potential or generation of another action potential. When all is done, the axon will return back to the resting potential. Sodium outside, potassium inside, both sodium and potassium gates closed, a negative 70 millivolt transmembrane potential. And it now is ready for the next action potential to be started. Okay. This figure is just putting all of those steps together on one figure so you can do a review of the action potential, starting with the resting potential and the leaky sodium gates, then hitting threshold, which causes the sodium gated channels to open wide and let the sodium run in, hitting positive 30 with the depolarization occurring. And at positive 30, the sodium gates close and potassium gates will open, so we can throw the potassium out. Now we are getting back down to near negative 70, but we're slow to close the potassium gates, so it takes all the way up to negative 90 millivolts, which is hyperpolarization. We close the potassium gates, and now we depend on the sodium-potassium exchange pump to pump sodium out, potassium back into the cell, so we return to the resting potential. And the resting potential now has sodium outside, potassium inside, negative 70 millivolts transmembrane potential, and both of the gated channels are closed. Now, the time after we hit threshold. As soon as we hit threshold, the action potential is gone. We now want to clean up our area so we can get it back to resting potential, so we can get hit by another action potential. Okay, now the action potential is jumping down and it's touching down on the axon, doing this over and over and over again. Okay. So the time right after threshold is called the refractory period. And this is the time the axon will normally not respond to any new depolarization stimuli. Okay, it's a cleanup time. So we're going to break this refractory period into two different sections. Absolute refractory is going to be from threshold, when it was negative 60 millivolts, to the positive 30 millivolt membrane potential. During this time, your sodium channels are open. So when we started the action potential, we had to open the sodium channels. So if they're already open, we can't open them again. They're open. So there is no way that we can get another stimulus occurring. So your sodium channels are open until inactivation of the channel ends. Then they can be reopened. Then they'll enter into the relative refractory period, from positive 30 millivolts to negative 70 millivolts. This is going to begin when the sodium channels have regained their normal resting condition, so they do have the potential of being opened. However, they don't really want to open yet. They want to wait until we get back to our resting potential before they really want to open. So if they are going to open, it's going to take another action potential. It's going to take a much larger stimulus than a normal stimulus. Okay. I say in class, usually think of it if you have a party. If you have a big party, and then when you are done with the party and everybody leaves at 2 in the morning, the last thing you're going to do is let somebody in if they come in at 3, because you're cleaning up and you don't want to have another party. But if somebody comes by around 7 in the morning, you still don't want to have another party, but they're got a good reason to have a party, you might actually let them in before you're ready to be set for the next party and you're cleaned up. Okay, now we're going to talk about propagation of the action potential. That is moving the action potential down the axon from the beginning to the end where the synaptic terminals are. Okay. This is going to be affected by two things. It is going to be affected by the degree of myelination of the axon and the diameter of the axon. Okay. Now, we're interested in the speed of propagation because we want it to go quickly for some of our information to get processed quickly. We talk about degree of myelination. You have unmyelinated axons with no myelin sheath and myelinated axons that do have the myelin sheath. With the unmyelinated axons, the action potential takes these little tiny steps and all the way down the axon. And each time it steps down, it has to do the whole process of going from resting potential to generating the action potential. So it takes a lot of time to slowly take baby steps down the whole axon. It is also energy expensive because you need to use ATP every time you do all that processing. This type of propagation is called continuous propagation. If we have a myelinated axon, the axon will be wrapped with myelin sheath. And you'll have an area of myelin sheath and a little area where there's bare axon, and that space is called the node of Ranvier. And then there'll be another chunk of myelin sheath followed by another node. The action potential is going to jump from one node to the next node, and then do the whole process. But now, instead of taking small, tiny steps down the whole axon, we're jumping, taking large leaps down it, and so it's going to go much quicker, and it's going to use less energy. This type of propagation is called saltatory propagation, and salt means to jump in Greek. Okay. When we're looking at the axon diameter, the larger the diameter, the quicker the propagation. So we have three different types of nerve fibers. Type A fibers have the largest diameter axon, about 4 to 20 micrometers, and they're myelinated. Those two things add up so that they can carry their action potential at about 300 miles per hour, which is really quick. And this is going to carry sensory information about things like balance and delicate touch to the central nervous system, and then the motor commands back out to the skeletal muscles. Type B fibers are smaller, 2 to 4 micrometers, but they're still myelinated. Their action potential is about 40 miles per hour. And type C fibers are smallest, less than 2 micrometers, and they are also unmyelinated, and their action potential travels at about 2 miles per hour. Type B and C carry sensory information to the central nervous system about general senses, temperature, pain, touch, okay? Not delicate touch, rough touch. And it'll take the motor commands to the smooth muscle, cardiac muscle, and the glands. So far we've talked about action potentials in the nervous system only, but there are muscle tissue that can be excited and pass on action potential along also. The differences between muscles and axons are a number of them. We're going to look at 3 of them. The potential difference is larger in the muscle cells than in the axons. Your resting potential in skeletal or cardiac muscle is about negative 85 millivolts, whereas in the nervous system it was negative 70. Threshold is the same, whether it's the nervous system or the muscles, that's negative 60. So they got to have a little bit more of a hit, more graded potentials for the muscles to hit threshold and start an action potential. Once the action potential does start, it will last longer in the muscle cells than an axon. Axon is about 0.5 milliseconds, skeletal muscle 7.5 milliseconds, and cardiac muscle 250 to 300 milliseconds. And last, despite the larger diameter of the muscle cells, which in neurons will speed up the propagation, action potential propagation is slow in muscles. Smooth muscle is a totally different structure and responds totally different than cardiac or skeletal muscle or the nervous system. Your smooth muscle does not have typical excitable membrane. It doesn't have a stable resting potentials, and the action potentials or do they produce action potentials similar to other muscle types. And changes in membrane potential is going to due to calcium ions rushing in, not sodium ions, which is the case with the axon and the other 2 muscles, skeletal and cardiac. We've now propagated the action potential all the way to the end of the axon, to the synaptic terminals, and now we are going to look at synaptic activity. This is when the nerve impulse is going to be transferred from the first cell to the second cell, the presynaptic cell to the postsynaptic cell. The presynaptic cell is always going to be a neuron. Postsynaptic could be a neuron or a muscle at a neuromuscular junction or a gland at a neuroglandular junction. Now we have 2 different types of synapse, an electrical synapse and a chemical synapse. The electrical synapse is relatively rare. This occurs both in central nervous system and peripheral nervous system, but is really extremely rare. And in this case, the presynaptic and postsynaptic membranes are actually locked together by gap junctions. So they're very, very close, and they're bound by those gap junctions, which, if you remember, are made up of proteins called connections. And these proteins will form a nice circular structure around a hole or pore, and then that pore will allow the electrical impulse to jump directly from the presynaptic neuron to the postsynaptic cell. Okay? Again, relatively rare. They're quick, they're efficient, they always get passed to the next cell, it always excites the next cell. In the brain, this can be involved in conscious perception of different things. The second synapse is much more abundant, and this is your chemical synapse. This is going to occur between neurons and also between neurons and the other cell types. And in this case, the presynaptic and postsynaptic cells don't touch. There's a space between them called the synaptic cleft. And when the action potential gets down to the synaptic terminal, it causes the presynaptic cell to release neurotransmitters, which are those chemicals that convey information to the postsynaptic cell. The neurotransmitters are going to move across the synaptic cleft, bind with the receptors on the postsynaptic cell, and then they will trigger some kind of response from the neurotransmitter. Now from the postsynaptic cell. The neurotransmitters may or may not bring the postsynaptic cell to threshold. Some neurotransmitters are called excitatory neurotransmitters, where they can cause depolarization, and if they hit threshold, they can promote the action potential. Some are inhibitory neurotransmitters, and they will cause hyperpolarization, therefore inhibiting the action potential from being started and then propagation occurring. Now, the neurotransmitters, their effect is not determined by the neurotransmitter themselves, but by the receptor. Think of it as a light switch. You can have a light switch with 3 switches on one plate, and the switches all look the same thing, but if you turn one on, it turns the light on. If you turn the other one on, it turns a fan on. So they're wired differently. Even though it's the same receptor, they get turned on, but they do control different things. We're going to talk about one neurotransmitter, acetylcholine. Okay, now, acetylcholine is so common and released at so many places that we call the synapses that release acetylcholine cholinergic synapses. They are going to be found at all neuromuscular junctions, which we talked about when we did skeletal muscle. There are many synapses in the central nervous system, all neuron-to-neuron synapses in the peripheral nervous system, all neuromuscular and neuroglandular junctions in the parasympathetic division, that is the rest and digest division, of the autonomic nervous system. Now, quickly to go over how this works, because we did it thoroughly in the muscle system, but we'll do it again. We're going to have an action potential arrive down at the synaptic terminal, and it will depolarize the synapse. Okay. Depolarization is going to trigger the opening of voltage-regulated calcium channels. This is new information we didn't talk about with the muscles. Okay. The calcium channels open, and the influx of calcium into the synaptic terminal is what's going to trigger the exocytosis of the acetylcholine from the vesicles. So the vesicles go over and fuse with the cell membrane because of the arrival of calcium. And when they fuse, they spit out the acetylcholine into the synaptic cleft. This will then move its way across the synaptic cleft and sit in the receptors over on the postsynaptic cell. And once they sit in the receptors, this is a calcium-gated channel. It is going to cause the presence of the calcium. Acetylcholine will cause the gate to open. That will allow sodium to charge into the postsynaptic cell, and that will produce a greater potential, possibly reaching action potential. As soon as this is happening, the ACH is quickly being removed from the receptors by acetylcholine esterase, the enzyme, and then it's going to be recycled either back to the vesicles, or it's going to be broken down and used for other metabolic activities. Now, there's always going to be some degree of synaptic delay. This is the delay between the time the action potential arrives at the presynaptic neuron at the synaptic terminals, and then the action potential makes its way across the synaptic cleft to stimulate the postsynaptic cell. This is going to be about 0.2 to 0.5 milliseconds. And this is because it takes a while for the process to occur to get the action potential down there, open the calcium gates, let the acetylcholine out, have it move across the synaptic cleft, bind with the receptors, and then open the gates. Okay. We can also see what's called synaptic fatigue, and this is due to intense stimulation when the presynaptic cell runs out of acetylcholine. And because so much of the acetylcholine is being released because it's getting hit over and over and over again by an action potential, it's releasing the acetylcholine so quickly, and it can't recycle it quick enough to pick it up and put it back into the vesicles, so we run out of acetylcholine. The synapse will then remain inactive even if it's getting hit by action potentials until the acetylcholine has been replenished. There are two ways that the neurotransmitters can have an effect on the postsynaptic nerve. They can have a direct effect on the postsynaptic cell, or they can have an indirect on the postsynaptic cell. A direct effect is an example of that is when the acetylcholine binds right on the receptor, which is one half of the gate, it causes the gate to open. So it's immediately, if you knock on the door and somebody opens the door, that's a direct effect. Okay. And that you can see with acetylcholine as well as other types of neurotransmitters. The indirect effect is when the neurotransmitter goes across and sits on the receptor, But the receptor doesn't open a gate. It instead triggers some type of chemical reaction within the cell, which then goes and opens a channel somewhere else located on the membrane. So with this example, if somebody comes and knocks on the front door and you look out and you see who it is, you say go around to the back, and you send your kid to open the back door to let him in, that would be an indirect effect. And this you can see with norepinephrine and dopamine, okay, as well as other types of neurotransmitters. There are many types of neurotransmitters. We've talked about only acetylcholine, but there are other ones. Examples are norepinephrine, which occurs in the brain in the sympathetic division of the autonomic nervous system, and this is definitely excitatory. Okay. Dopamine is a second neurotransmitter, which is in the brain. It can be inhibitory or excitatory depending on how the receptors are wired, and a decrease in dopamine will cause an abnormality in your brain activity, which will result in muscle twitching. With Parkinson's disease, it's a problem with the dopamine neurons producing enough, and that causes the muscle activity that occurs. Third one I'm going to mention is serotonin. This is important for emotional state. It's usually an inhibitory, although it can be excitatory at times, and is found in the brainstem. And serotonin is now one of the chemicals in a lot of medications now for anxiety. Okay. And the fourth one is called GABA. Its name is gamma aminobutyric acid. This is in the brain, and it functions in anxiety reduction and motor coordination, and is another inhibitory neurotransmitter. Okay, now we're down to information processing. This is when that postsynaptic neuron receives the incoming information from the presynaptic neurons. Now, in reality, a single neuron can receive input from thousands of synapses at the same time. They'll have neurotransmitters of different type coming at them, or maybe the same one, but the receptors are wired differently. So some will be excitatory, while others will be inhibitory. And the net effect of all these incoming potentials is basically like math. You got to add it up. If this one takes me from -70 and depolarizes me to -65, the next one hyperpolarizes me back to -75, and you keep adding it all up and adding up all these incoming graded potentials. If they take the cell to threshold, then the action potential will start. Now we have different types of potentials here on the postsynaptic cells. These are the graded potentials. They can be excitatory postsynaptic potentials. These are ones that are depolarizing the membrane, or inhibitory postsynaptic potentials, which are hyperpolarizing the membrane. And when we add these all up, it is called summation. And we add them up, and that will, if it hits threshold, then the action potential starts. If you add it up and it is not hitting threshold, it is still more negative than threshold, then no action potential will be generated. There are two ways we're going to sum up or add up these incoming potentials. We can do temporal summation, and this is when you add up all the inhibitory and excitatory postsynaptic potentials that are at one single synapse over a short time period. Temporal means time. So if you get a hit over and over and over again in the same spot, but with different potentials, and you add all of those up, you see if that potential reaches threshold and if an action potential will get started. The second type of summation is spatial summation. And this is when we're going to add up all the inhibitory and excitatory postsynaptical potentials coming in simultaneously at the same exact time, but on different locations on the neuron. Again, if they add up to threshold, the action potential will start. Neurons can undergo facilitation, and this is when the neuron is brought closer to threshold. And it said it's facilitated. So if you get one graded potential and it takes you from -70 to -65, the next graded potential doesn't have to be as large to push it over the edge and hit threshold. So the larger the degree of facilitation, the smaller the additional stimulus has to be. So if I take it from -70 to -65, I get hit by one that pushes me over to 6. I need to have a decent sized stimulus. If my first one took it to -70 to -62, I can need a smaller stimulus to then push me over the threshold to start the action potential. In the same thing, we can have inhibition and facilitation on the presynaptic neuron. The presynaptic neuron can be inhibited, and the way that happens is when it is inhibited to release its neurotransmitters. An example is GABA. GABA inhibits the opening of the calcium gates in the synaptic terminal, and if the calcium gates don't open and the calcium doesn't go in, then the neurotransmitter is not going to be released from the presynaptic neuron. The presynaptic facilitation is when some type of activity will increase the amount of neurotransmitters that's going to be released when the action potential arrives. Serotonin will keep the calcium gates open longer. Thus, the stimulation period is extended, allowing more neurotransmitters to be able to get out and move across to the postsynaptic cell. Your neural function is going to be affected by many factors, and most of these factors are the ones we try to keep in homeostasis. Okay. The first one is pH. If your pH normally is 7.35 to 7.45. If you get a rise in the pH, then the neurons are going to get facilitated, which means they'll work more. Okay? A pH of 7.8 will cause severe convulsions, and your action potentials will be produced spontaneously. A decrease in the pH, and the neurons will be inhibited. With a pH of 7.0, the AP generation will slow so much that the nervous system shuts down completely. The second thing that will affect neuron function is your body temperature. Increase in temperature, the neurons get excitable, and we have convulsions and hallucinations. This is what happens when you get a high fever. A decrease in body temperature, and your neurons get inhibited. You become lethargic and confused. Changes in ionic composition of the extracellular fluid will also have an effect on the neurons and how well they function. And it can either be inhibitory or facilitate the neuron depending on which ion is involved. We also need to be very concerned about our nutrients. If we don't have enough nutrients, the brain will not work properly. Your brain is totally dependent on a continuous source of glucose for cellular respiration. It does not store any glycogen in the brain. So if you're not getting enough glucose or converting molecules to glucose, the brain will have a hard time functioning and doing cellular respiration and making ATP. Also needed for ATP is oxygen. Okay? Your brain only does aerobic respiration. If your circulation is interrupted for only a few seconds, the neurons will be injured. Okay? And if it's for longer than that, you'll have neuron death. Now, your brain is only 2% of your body weight, but it uses 18% of the ATP that is produced under resting conditions.